All right, today we're going on a little journey. We're going to follow a single super important atom, sulfur, as we unpack the metabolism of cysteine and cysteine. It's a really cool story of how it's made, what it does, and yeah, what happens when things go a little sideways.
So let's just dive right in. I love this quote because it really sets the stage perfectly. Cysteine might seem small and simple, but its impact is just huge. It's involved in everything from the literal shape of our proteins to how we fight off cellular damage.
Okay, let's break down our two main characters here. First, you've got cysteine. It's a single amino acid, a monomer. Now, it's technically non-essential, but there's a really important catch. Our body can only make it if we've got enough of another amino acid, methionine, coming in from our diet. Then, when two of these cysteine molecules get oxidized, they link up and form cysteine. That's the dimer, connected by a super strong disulfide bond.
So that begs the question, right? How do we get that cysteine in the first place? Well, that brings us to a process called the transsulfuration pathway. You can really think of cysteine synthesis as a story of teamwork. It's like this biochemical relay race that connects the metabolism of two other amino acids, methionine, which is essential and brings the sulfur to the party, and serine, which provides the carbon backbone. So let's walk through it. Step 1. It all starts with methionine, which we have to get from food. Our body converts it into an intermediate called homocysteine. Step 2. That homocysteine then links up with serine to form cystathionine. Now this step is absolutely critical, and it needs an enzyme called cystathionine beta-synthase, and importantly, its helper, vitamin B6. And then finally, step 3, another B6-dependent enzyme comes along, cystathionase, and it chops up cystathionine to give us our final product, cysteine. The big takeaway here, no vitamin B6, no cysteine. It's that simple. Okay, so now that we've successfully made some cysteine, let's look at all the different hats it wears in the body. I mean, this is where it gets really, really versatile. So now that we have it, what are its essential jobs? What does it actually do? Cysteine is a serious multitasker. In protein synthesis, its little sulfur atom forms these incredibly strong disulfide bridges that give proteins their stable 3D shape. You can think of it like molecular glue holding everything together. It's also a key ingredient for making glutathione, which is basically the body's master antioxidant. On top of that, it's essential for coenzyme A huge player in metabolism. It's a precursor for taurine, which we need for bile salts, and it can even donate sulfate for detox reactions. Wow, that's quite a resume, right? So we've mentioned cysteine and its oxidized twin, cysteine. Let's really dig into that dynamic relationship now, the cysteine-cysteine connection. It's what we call a reversible redox pair. Okay, here we can see the two forms right next to each other. Cysteine is the reduced form. It's a single molecule ready to give up electrons, and you find it mostly inside our cells, in the cytosol. Cysteine, on the other hand, is the oxidized form. It's two cysteine molecules all linked up after they've lost their electrons, and it's much more common in the oxidizing environment outside of our cells. This back and forth process is just crucial for keeping our cells' redox state in balance. All right, now for the part that every medical student really needs to nail down. Let's jump into our clinical crash course and see what happens when this whole beautifully balanced system goes wrong. This slide is super high yield for exams, so listen up. With homocysteinuria, the root problem is a synthesis failure. That enzyme, cystathionine beta-synthase, is defective. So you can't make cysteine properly, and homocysteine builds up in the blood, which can lead to some really serious vascular problems. Now, with cysteinuria, it's a completely different issue. It's a transport failure. The kidneys just can't reabsorb cysteine like they should, so it gets super concentrated in the urine, forms crystals, and bam, painful kidney stones. So the key difference to remember, homocysteinuria is a problem making cysteine. Cysteinuria is a problem transporting cysteine. All right, let's bring it all home. We're going to do a quick summary and then a rapid-fire quiz to really lock all this information in your brain. So to recap the whole pathway one last time, nice and simple. We start with methionine. That becomes homocysteine, which turns into cystathionine, that yields cysteine, which can then be reversibly oxidized into cysteine. See? A really simple, elegant flow. Ready for a quick quiz? Okay, first question. Which vitamin is absolutely essential for making cysteine? That would be vitamin B6, specifically its active form, pyridoxal phosphate, or PLP. Don't forget, it's needed for two of those key enzyme steps in the pathway. Next up, what is the fundamental relationship between cysteine and cysteine?
Yep, cysteine is the oxidized dimer that's formed from two cysteine molecules. It's the monomer versus the dimer, reduced versus oxidized.
Question number 3, let's go clinical. What is cystinuria?
That's the one. It's a genetic defect in how the kidneys reabsorb cysteine, and it leads to kidney stones. Remember, it's a transport problem, not a synthesis one.
And for our final question, which incredibly important antioxidant has cysteine as one of its key building blocks?
Glutathione. That's our body's master antioxidant, and cysteine's role in it is absolutely non-negotiable.
And just to leave you with one final thought that ties this all together so nicely, here's a great mnemonic to remember. Methionine supplies sulfur. Cysteine sustains structure. It just perfectly captures the whole story, doesn't it? Methionine brings in the key ingredient, and cysteine uses it to build and to protect. And that's our deep dive into the sulfur story. Thanks so much for joining me.